Let's come back to the topic of protein digestion. So the main concept about protein is that we need acidic pH for breaking these complex protein chains into these monopeptide chains, right? And we eat diet, and our diet is mainly compromised, not compromised. Our diet has mainly polypeptides in it, and we have to break down these polypeptide into monopeptide. That is digestion simplified, right? Now for this to happen, we need pH of around 2, and we need to convert these pepsins into pepsinogen. And this pepsinogen, we need these enterokinases, right? These enzymes to convert this pepsin into pepsinogen, and we need a pH of pH 2. So that's the whole story, right? Let's start by singly understanding each and every concept of it. Okay, the first thing is that we need these endopeptidase. What are these endopeptidase? These endopeptidase are these enzymes, are these catalysts that act on these pepsinogen, right? Not they act on the pepsin, and they just activate this pepsin, right, into pepsinogen, right? So we have these endopeptidase, we have this exopeptidase. Now endopeptidase, they act on the inner region of the peptide bonds, and exopeptidase act on the end region, the external part of the peptide bonds, right? So endopeptidase, they work on the inner region. For this is my peptide chain, right? There is this COH bond, and then there is H2H bond, right? And this endopeptidase work on the inner side and breaks down. We get H2H and COH here, and then again we get 2 chains right here. And exopeptidase breaks on the outside here, on the outside. Now, right, when exopeptidase work, we get these smaller bonds and these one big chain. So let's try to understand why we need these specific endo and exopeptidase. Why can't we have just one peptidase? Because endopeptidase just increases the number of ends available for exopeptidase to act upon. Okay, so for the complete breakdown of these complex polypeptide chains, we need both. Okay, so that we have the whole complete breakdown of whole amino acid, because it's like an assembly lane, right? Why not have just one enzyme that does all the activities? Because assembly line design, because it's more efficient, it's more in control. If something goes wrong, something goes rogue, we can have more regulation on them, right? Okay, so that's endopeptidase and exopeptidase, right? These are 2 enzymes that are needed to convert these pepsin into pepsinogenase, and then the further digestion happens, right? So our stomach makes this pepsin, our pancreas make trypsin, our small intestine makes 2 type of these enzymes called chymotrypsin and carboxypeptidase. Now I have used these 4 enzymes name, right? Pepsin, tripepsin, trypsin, trypsin, whatever you want to call, chymotrypsin and carboxypeptidase, right? Now other thing is that now these are just type of endo and exopeptidase, right? So pepsin is a type of endopeptidase, trypsin is also a type of endopeptidase, and chymotrypsin is also a type of endopeptidase. So all 3 of them are produced by the body, and they work on the inner chain, right? They just increase the number of small chains so that exopeptidase can work efficiently. And this one called carboxypeptidase is an exopeptidase. It works on the external one, right? Okay, so this is this is clear. Now pepsin is released by stomach because we need pH of around 2. Duodenum, right, the closest small intestine area close to stomach, produces trypsin, chymotrypsin and carboxypeptidase, right? So duodenum produces 2 endopeptidase and 1 exopeptidase. Okay, intestine overall generally produces this thing called aminopeptidase, and this is also a part of exopeptidase. Okay, so carboxypeptidase and aminopeptidase both work on the end region of the whole peptide chain, right? So I hope this little concept is clear about the peptidase and endopeptidase and these. Okay, now these forms of enzymes they are present in the form of zymogen. Of course, because if these enzymes are present in the form of active enzymes, they will eat themselves up, because protein is present almost everywhere in the body. So imagine these enzymes, they get activated while they are inside the pancreas. Then you will have this pancreatitis, and this pancreatitis will digest the pancreas itself. Once the pancreas is digested by itself, by its own juice, you will have something called as liquefactive necrosis. Okay, so that's one another point, right, to understand. And so we need to keep these enzymes in a very inactive form, and that's inactive form is called zymogen, and it prevents autodigestion of pancreas, of course. So activated in small intestine. How they are activated, how these things, how these these endopeptidases are activated. Well, there's this trypsin, right? So trypsinogen that is produced by pancreas enters in the duodenum, right? And then we have this enterokinase, right? These cells, enterokinase cells, present at intestinal brush border, they convert this trypsinogen into trypsin, okay? And then trypsin is the main trigger for conversion of this whole thing. So once we digest this polypeptide, we get the end result is dipeptides and tripeptides, right? And that's the whole thing happening, right? So one really important hormone is this, not hormone, this, and this enzyme is trypsinogen, right? Okay, let me go back to this chart where, wait. Yeah, this chart is super important, right? Activation of zymogen in small intestine. The trigger for activation of these enzymes is enterokinase, okay? Enterokinase or peptidase, you can call them. We call them enterokinase or endopeptidase or peptidase, whatever you want to call them. And this is the initial trigger for everything, okay? Once this enterokinase, right, this protein, it comes from duodenal brush border in small intestine. So duodenal brush border, they are really rich in a lot of sensing capabilities. They sense if we have some sort of amino acids. Once they sense that we have amino acids, they will release these enterokinase, and these enterokinase will act on trypsinogen. Now this trypsinogen will be this zymogen called trypsinogen will convert into trypsin. Now trypsin is really activated. Now trypsin is gonna digest a lot of protein, and it's gonna positively influence procarboxypeptidase, which is an exopeptide, and this chymotrypsinogen, which is an endopeptide, right, peptidase, is going to activate them, and then we're going to get chymotrypsin, and then we're going to get carboxypeptidase, right? So trypsin is the number one factor for these activation. Again, carboxypeptidase is an exo, chemo is an endo, okay, and eventually protein digestion is going to happen, okay? So that's the story for the protein digestion right there. Okay, so once we have digested all the protein, right, I hope we can skip that. Now we can understand this whole part, right? Now we are left with these form called dipeptide and tripeptides after the digestion of polypeptides, right? So absorptions. Now the absorption happens from 1 to 4. I have labeled it. So first is these peptide transporter called peptide transporters. Second are these sodium proton exchanger. Again, it's exchanging. Exchangers mean they just exchange one atom for another one. Third is these cytoplasmic proteases. Proteases mean it cuts the protein into 2 parts, okay, or maybe 3 parts. It's just a proteases, okay, it cuts down the proteins. And fourth is a basolateral amino acid transporter. So amino acids, these are just proteins, and transporter is just transports, and basolateral means on the blood side, right? So this is the blood basolateral side, this is apical side, this is the lumen, right? And what happened here is that we have these peptide transporters. They transport the dipeptide and tripeptide inside the cell. Once the cell has these dipeptide and tripeptide, they are acted upon cytoplasmic proteases. Once these cytoplasmic proteases are acted upon, they cut the dipeptides and tripeptides into single amino acid chains. Now these single amino acid chains are further just pushed inside this basolateral amino acid transporter, right? Now you must be thinking, what is the work of this singular channel, singular exchanger? Why does it even exist here? Because here we can see, conceptually we don't need this one, right? But you are not right here. I'll tell you what really happens, what really goes on. First question is that which cell is this? This cell where all this happens is enterocyte, right? The absorptive epithelium cells lining these small intestinal villi, okay, mainly jejunum and ileum, okay. Now these cells, these are like superpolarized means they are highly charged, okay. So the peptide is absorbed via pepti, right? Pepti is the channel. It absorbs amino acids, dipeptide, tripeptides, nine. And I'm just gonna repeat myself again. All of this thing happen. The cytoplasm proteases, it drops, it cuts down the amino acids, right? Now for these for these to enter into enterocytes, amino acids use these sodium dependent amino acid transporter. These are co-transporters, okay. And dipeptide and tripeptide uses this H plus dependent co-transporter called peptide. So this is the reason why we need this H plus and Na, okay. So let me just write it, okay, because it's really important. The peptide, wait, okay, the first point is amino acids, they use Na dependent acid transporter, okay. It's a dependent transporter, okay. It's co-transported with sodium, okay. And di and tri peptides, they use proton dependent co-transportation, okay, transport, and this co-transport is called pep-T1, okay. So pep-T1 brings these peptides inside the cell along with this thing called sodium. So this is the reason why we need this exchanger, okay. This is the reason. That means what that means that as these dipeptides they come inside, the protons also flood in, making these cytoplasm slightly more acidic. Now the problem here is that there are too many protons inside the enterocyte. If the cell becomes too acidic, the transport of these enzymes and then this transport of this peptide will fail, because this is a pH sensitive process, right? So enterocyte must export this excess proton. That is because of the peptide channels. Now the solution for that, now the nature has given this solution, is that we were given this sodium proton exchanger. The other name for these is NHE3 channel, okay. And these channels, now they are exchangers, they are not channels, and it takes sodium in and it pumps proton outside the lumen, and it achieves two things. First, we are removing the protons. Second, it brings the sodium, which will be later used so it pushes a lot of sodium in. Now this sodium, it will be later be pumped out basolaterally via these sodium potassium ATPases to maintain this sodium gradient. So if you were wondering what's going to happen with this sodium, well, it's just pushed in the basolateral side. So it's a beautiful coordination. This is why God or evolution, whatever you want to think, gave us these peptide transporters with this Na and proton exchangers. And then on the basolateral side, we have this amino acid transporter which pushes amino acid, and it pushes this excess Na in the blood, right? And then we have this cytoplasmic proteases. It just cuts or maybe split dipeptides and tripeptides into amino acid for release into blood. So sodium proton exchanger is not random. It's a part of that loop that powers nutrient absorption while keeping the pH stable of the cell, right? Keeping the pH stable. Okay, so the amino acids are in the blood now. Once the amino acids are formed in the cytoplasm, they leave, right? And I think they are these sodium dependent or sodium independent pathways, right, where the amino acids can leave inside the blood. So I hope we understand this whole absorption, right? Okay, I've written somewhere that why we need cytoplasmic proteases, right? Why we need, I thought about it because I thought that why don't they just appear in the lumen itself, and we will save this peptide and this exchanger work. We can just directly produce them in the blood. And it turns out that these proteases can damage normal cell mucosa, and they will digest the living cells. So that's why we cannot have them in lumen or in the blood. And if they are inside these cytoplasmic, if they are intracellular present, they are controlled well. They controlled contained environment, they are inside this environment, and small environment equals to small ATP demand, because if they are in a bigger environment, they will need more, more, because they will be metabolically highly active. Okay, we want them to be not so ATP demanding. Okay, because we want the absorption of protein to be really, really, really efficient. Okay, so that's the concept. Let's talk about vitamin water solution. Really simple topic. So this vitamin water absorption, right, it depends upon how big and bulky the vitamin is. Small and polar, they ride sodium. Big and complex, they use receptors. And fat soluble hide inside the micelles. Okay, that's the whole concept of vitamin. So fat soluble are A, D, E, K. These are fat soluble. They can hide in micelles, right? Micelles are these jejunum. They are formed in jejunum. They are just they are just bile salts, inverted, and they are vehicles, right? I hope you understand micelles. If not, okay, let me show you micelle again. This is a micelle, and this is how it is formed in the jejunum, and it has one hydrophilic shell, so that it can safely be soluble in water, and it has one hydrophobic content. That's the fat ADEK, right? ADEK, free fatty acid inside, cholesterol, these things inside. So the fat soluble vitamins are absorbed inside micelle. Big and complex materials, they use receptors, and small and polars, they ride Na. Okay, so what can really happen here is that a saturation can happen at Na. So we have limited number of Na, right? We don't have unlimited number of Na. Just imagine if these small and polar, these small polar vitamins, small polar nutrients, they overpower these carriers. If they are more, these small polar things are more, and the Na carriers are less, then we will hit this plateau on transportation. So just know that. Okay, so vitamin absorption. So fat soluble vitamins absorb along the micelles, I know. Water soluble, such as vitamin B complex, vitamin B complex absorbed by sodium dependent co-transporter. They ride the sodium, I know. And except vitamin B12 and folate, that is B9, right? And they transport independent of Na, because both B12 and B9 are large. These are large complex, right? And too complex. They need specialized carrier transport. Okay, so I hope we knew that. Okay, let's talk about B9 first. So B9 is called folate, and most of the absorption is done via jejunum as monoglutamate form after this deconjugation, right? So we have two transporters for folate. We have PCFT and RFC. These are two transporters, right? Okay, let me go again and explain you B9 folate absorption again. Okay, so this happens at jejunum, right? Now just listen to the story, right? Now the main site is jejunum, mainly proximal small intestine. What really happens is that folate comes to duodenum and it moves to jejunum, and at the brush borders of these jejunum and these duodenum, the apical membranes, right, it faces the lumen. Apical membrane faces the lumen, and basolateral faces the blood, right? So at the brush border of these enterocytes, the absorption of this folate happens. Now the story is this, step by step, listen to it. Dietary form and the first problem, right? Now what's the first problem? The first problem is that polyglutamate cannot cross membrane. What is polyglutamate? Polyglutamate is the form of folate that is present in the dietary forms. Okay, so dietary folates such as plant folate, right, and they exist in a form of polyglutamated folates, right. Multiple, poly means multiple, glutamated means glutamate, and folates mean polyglutamate residues attached with each other. It gives us polyglutamated folates, right. So these polyglutamates, they cannot cross this membrane because they are like big, and they are like big complex molecules. They cannot cross this bilipid layer. So we must reduce these polyglutamates to this monoglutamate level, right. Now this reduction, it happens, how it happens. So on the brush border, right, there are these FYGH, right, often called as conjugases, okay, or maybe Y-glutamyl hydrolase, right. These enzymes remove this extra glutamate residue, and it produces this folate glutamate. Now this folate monoglutamate, right, it's also called 5-methyl tetrahydrofolate, 5-MTHF. Now this 5-MTH is reduced for food. Folic acid is oxidized from supplements, right. So the uptake, right, uptake means just absorption. So the absorption of this 5-MTHF, right, level, this form, 5-MTHF form, is done via these PCFT cells. Now the PCFT is protein coupled folate transporter. Simple name, protein coupled folate transporters, right. Now it's I hope you understand this. Now PCFT is a proton coupled transporter. Now it is co-transports folate with proton. So it works best at slightly acidic environment, right. So the microclimate is acidic. Okay, so microclimate pH at apical surface, that the lumen surface, is often more acidic, right. So that's why these PCFT cells are present on the lumen side, right. And again, if the lumen is acidic, it means we have abundance of protons, and now we can carry this folate with proton. Okay, other route is that RFC, right. Now this RFC, this RFC, right. RFC is this folate receptors, right, or we can call them this thing called we can call them FR alpha. These are multiple names. What really happens here is that in some tissues, right, under some stress or under some condition, the folate can also be taken up, right, by these RFC receptors, right. And this is a receptor mediated endocytosis of folate. And most importantly, it happens in placenta, right, and even in choroid plexus where the CSF is made, right, and in some epithelial cells, right. And this PCFT is the main pathway. PCFT is the main pathway for intestine, and RFC is the main pathway for placenta and choroid plexus where the CSF is formed. Just remember that. Okay, so this is the story, RFC. So I hope we understand this, story. Okay, so that was folate. They don't need this sodium gradient. They usually need H plus gradient. Okay, PCFT cells. Okay, B12 story. Okay, for B12 story, I would. Okay, let me just. Okay, so B12. Where is B12? Here is B12 story. So this is the B12 story. We eat B12 in the diet. The stomach releases something called as HCl, acid, and pepsin, right? Now this pepsin and HCl frees the B12 from a food. Now B12 binds with this R protein that is present in the saliva. Now this complex is really important because HCl will keep on destroying shit, and we don't want our B12 to be destroyed. So that B12 isn't destroyed from HCl. That's why B12 binds with R protein. Now there's something called the intrinsic factor, right? Now this intrinsic factor now enters inside the chat. Now this enters in the duodenum. Now, right, this intrinsic factor enters inside the duodenum, right? And this pancreatic protease, it digests the R protein from the B12. Now the B12 and the intrinsic factor are free. to meet with each other, right? They meet with each other, they form a complex, and they travel to ileum. Now, at the terminal ileum, there's a receptor called Cuban receptor. Now, this complex is endocytosed via this Cuban receptor, and it is endocytosed inside these enterocytes, right? Now, this complex is inside this enterocyte. Now, the B12 binds with something called as transcobalamin 2. Now, this is a carrier for B12, and now this B12 is free to move inside the blood. So that's the story of B12 in short. I hope you'll understand this. So that's the story of B12 fast. So now we know B12, now we know B9. Okay. Okay, so B12 really works via intrinsic factor, via our protein, and via something called as transcobalamin 2. Okay, so we have three things to take care of B12. And for folate, we need these PCFT and RFC channels, right? And PCFT is proton dependent and RFC is not. Okay, so that's the cool. Water and absorption. So total water content in GIT is around 9 liters, right? That's a lot of water. Water reabsorbs around 8.8, and around 200 ml is lost in the poop, in the stool. So maximum water is absorbed in the jejunum. That's important to remember. Now, this is really important. Why? Because most glucose, amino acids, sodium and chloride is reabsorbed here at the jejunum. Now, by the time the mixture reaches the large intestine, 90% of water is reabsorbed. I'm just giving you some facts so that you can remember, you can, generate some image in your mind, and you can think, oh, jejunum is this lover guy who loved this, this sodium, chloride, amino acid, glucose. So just remember that. Okay, so that was vitamin, water, and protein. So we are done with this thing as well. Next video, we're going to talk about iron absorption, which is amazingly simplified and it's so easy, and it's gonna build your concepts for pathology to understand about. If you understand the iron absorption well, you will never have problems understanding anemia. So I hope that says how important the iron absorption mechanism is.